Hey, how's it going?
Hey, Carl, what's happening?
Not much. No, I shouldn't say not much. It's been really busy, but
I bet.
just hanging out at home. So, how about yourself?
Oh, it's going good. A lot happening as we talked about last week. With this, would I be able to record this as well?
I'm not sure you can. I can record it. You can maybe record the audio, through any device you have there. But
Oh, okay.
okay, but I should be able to record this. It's recording right now.
Okay, terrific. Terrific. So, all right. Well, I guess we'll get to it. I don't want to take too much of your time, but essentially, like I said, so I kind of gave you a rundown of what my what our situation over here has been for the last week, and that just brings up a lot of questions that we're getting from very very similar questions that we're getting from everybody else.
Mhm.
So number one, introduction. Well, who is Carl? Credentials, experience, all that kind of stuff.
Sure. So I can tell you a little bit about myself. I'll kind of go back in time, way back in time. So my background is basically I'm a molecular biologist by training. Actually got my undergraduate degree at UCLA in biochemistry, and then did my PhD over in Oregon in molecular biology, and did my postdoc in molecular genetics at Stanford. And right after I did my postdoc, I actually went right into industry and would start to engage in a career in diagnostics with a strong focus in molecular diagnostics. Initially worked for a company called Applied Biosystems, now part of a very large corporation called Thermo Fisher. And there I worked on detection of various types of pathogens that are in food and environmental samples. So food safety, environmental safety types of applications. Later, I went to a company called Boehringer Ingelheim, worked on actually developing assays for various types of viral pathogens, human viral pathogens. For instance, a human papillomavirus that's involved with cervical cancer.
Yeah.
And then after that, the company was acquired by another company called Roche. That's the largest diagnostics company in the world. And there I started working on what's called microarrays, basically to figure out, whether people have certain variants in certain genes that are encoding enzymes in your liver that metabolize drugs. So this is a very important and sort of an area that's on the rise right now in terms of figuring out, whether people are able to metabolize certain drugs, or maybe they metabolize them too quickly or very slowly. But it helps to, down the road, it will help to better understand whether people should be given certain medications, or maybe they need to be dosed in a different manner than the average person.
So it really points in a direction
Wow.
for strategy for therapeutics.
Absolutely, yes, yes. And they go hand in hand with therapeutics. And in essence, this is something that could be done, when you're born. For instance, when a person's born, you can get types of genotypes, as they're called, and then you can have that for the rest of your life, so that, as part of your medical record, that you can then, that the healthcare professionals can then use to determine, what is the best course of treatment for you for whatever condition they're trying to manage. Then I worked for other companies. I came to Arizona a number of years ago, and I've actually been at Arizona State for the last 16 years. But I've also still stayed in industry in terms of consulting and doing some side jobs as, in various roles for small companies. And that brings me to today. And in fact, the last 5 or 6 years now, I've been actually part of a program, the Biomedical Diagnostics program within the College of Health Solutions at Arizona State University.
That I went through.
Right. So, Jason is a prime example of some of the really good graduates that we get from the program. And it's really a program that is meant to develop the future leaders in this field. So that's what I've been doing of late. And, certainly things that have been going on recently with this COVID-19 situation has, it's really been tough to deal with it and, to hear all the stories, but it's also a time for someone like myself that it's just a very interesting crisis situation and how it's been handled and how people are, responding to it and things like that, which I think we'll be talking a little bit more about, at least in the area of diagnostics.
Absolutely.
Interview.
Absolutely. Yeah. And so, I guess getting into, a lot of the questions that I've been asked, as we went through our own, personal brush with this, with regards to COVID, what kind of testing is being done? How far along is it? How accurate is it? How does it work? And I know this is a broad question, so, but, and then what does it mean and what are the implications of all that testing?
Well, I could give you probably a lesson that lasts a couple of days on this, but
Yeah.
sort of the very short version is that there are lots of tests out there. In the world, there are probably on the order of 450 or more tests out there to basically look for COVID in one way, COVID-19 in one way or another. In terms of diagnostics, in terms of determining whether you have COVID-19 or not within your system, within your body, the most common tests are what are called nucleic acid tests.
Mm-hmm.
This is where basically they do some, a nasal swab. So they take an extra long Q-tip and stick it way up your nostril and jimmy it around there. And then they take that and then put it in a tube. And then what they're hoping, not hoping, but what they're trying to find is basically the virus itself. And if the virus is there, then they prepare it so that they can release what's called the nucleic acid, the genetic material for that virus. And in this case, the virus, you have really two types of genetic material that can be out there. You have DNA or RNA.
Mm-hmm.
This type of virus is what's called an RNA virus. So what you have to do is you isolate the RNA, which is tricky to do because RNA is very fragile, much more so than DNA. And then what you have to do is you have to take that RNA and convert it to DNA. And that requires a special enzyme to do all that type of stuff. And then after you get into DNA, then you can do something that's actually very commonly used in this field. It's called PCR, polymerase chain reaction, which can amplify that DNA sequence or the DNA sequence of interest billionfold in a matter of less than an hour. It depends on what your protocol is. And then once you have that, then you can either detect it directly by looking for a fragment, using certain techniques, or you can have dyes that can have it light up, for instance, in a test tube, for instance, and be able to see it. But there's variants to how you detect it. But basically what you do is you amplify it and you detect it. If it's there, then that says that you've been infected with COVID-19, okay? So that's the main test in terms of diagnosis. And there are many tests out there right now. The FDA, the Food and Drug Administration that oversees the regulatory side of these tests, normally you'd have to go through extensive validation studies and things like that. But because of this situation with COVID-19 and there's the very strong sense of urgency, they have what's now called an EUA, so Emergency Use Authorization. So you still have to provide some data, but you can get it fast tracked and you can get it approved in the matter of a week, basically.
Okay. All right. And what about antibody tests? Are those, tests for some things via, antibodies, which, I'll let you explain that.
Yeah. So antibody tests are definitely becoming much more important now because now we have a lot of tests out there that can detect whether people are infected. What we don't know all the time is have people been infected but maybe have never had any symptoms or have very mild symptoms, such that they thought maybe they just had a cold and things like that. So those are people where they might be fine now, and you test them and see, have they been basically exposed to COVID-19? And so what happens is if COVID-19 gets into your body, there is typically what's called an immune response where you start to generate antibodies. This takes some time to happen. So antibody tests are not good for early detection and diagnosis of a COVID-19 infection. But what they can do is they can tell you whether a person has been exposed. And eventually it will tell you, what level of exposure or did you have? And also at what stage are you at? Because right now you can detect two different types of antibodies.
Okay.
Things that you hear once in a while, you hear IgM and IgG. These are, IG stands for immunoglobulin. Basically, it's just an antibody in a type of antibody. The IgM is something that's used when when you first get infected. That's the first type of antibody that comes up. It's part of the memory system, and it's something that can trigger a much larger response down the road. And that larger response results in the IgG. And so it's the IgG that potentially can give you immunity, at least for some period of time, to COVID-19. What people...
Oh, okay. So it's the IgG. I've kind of heard that. I thought it was the IgM actually. That was like kind of the long-term one, but that's actually a short-term one.
Well, it's a short-term one, and they're not present in large amounts.
Yeah.
And so, what happens is that if you trigger a cell that has the IgM on the surface and it binds to it, then basically it can proliferate, and then it can make a lot of IgG. Okay. And so that just says that your immune system has kind of gone through the process of mounting a significant immune response to that, in this case, to COVID-19. The thing that we don't know yet, or I should say, I don't know right now.
Yeah.
There might be some people that do know, but we don't necessarily have a test that can say, okay, you're positive for COVID-19 IgG, but and also we know that you're going to be immune to COVID-19.
Yeah, that's kind of what we were, trying to, get to in terms of implications is, two things is like, will you have natural immunity? And number two, at what point are you contagious or not?
Yeah, well, so right now, I think the thing is if the diagnostics, so again, the nucleic acid test says that you have COVID-19 in you, then you're considered to be, at this time, the standard is saying that you're infectious, or you can be in an infectious state. If you are COVID-19 negative in terms of the nucleic acid test, but you're positive in terms of antibodies, the assumption right now is that you are not infectious, and that you had been exposed to COVID-19, and that you may have immunity towards it. And more work needs to be done to really establish whether, what is sort of the algorithm that they have to put together to say, if you have certain antibodies against certain, what they're called antigens or markers, COVID-19, that would say that you have the right antibodies to mount basically an attack on any kind of COVID-19 that might come into your system from, say, a later infection or an event that could infect you, but now you have this immune response that can basically get rid of it.
Okay.
It's all being worked out. One thing to be very concerned about with these COVID-19 antibody tests is that because of the emergency use authorization, sometimes the data that is out there is not necessarily fully complete, and we don't know whether the antibodies are really specific for COVID-19, basically, or for.
Oh, okay. So they aren't that specific either.
They may not be. Now, some of them are, but some may not be.
Because I thought maybe their sensitivity was low because, they do say they're about 85 to 90% accurate, which is, pretty low.
It is. And the thing is that coronaviruses are actually a pretty common virus out there. It usually, well, one of the results is that certain other types of coronaviruses can give you basically a common cold.
Yeah.
And you can develop an immune response to that version of the coronavirus. So what you have to make sure, both with the nucleic acid test as well as the antibody test, is that it's truly specific for COVID-19. Okay. And not another one. And for instance, I have a student right now working on a project to develop a test, not for COVID-19, but for the MERS-CoV, so the MERS coronavirus, which is a Middle Eastern respiratory basically virus that has caused a lot of problems a few years ago, and actually even in the Middle East primarily. But that's a different coronavirus, and has a different mode of action. It's not quite as terrible as COVID-19, but it's something to be concerned about, especially if you're in the Middle East, for instance.
Okay. Well, that's also, I've heard that there's some different strains, even this virus, like from different from China to New York to Italy and here and Iran and all that. Is that accurate? Are they kind of different? Is there?
Yeah. So one of the things about viruses in general, and it kind of depends on the virus, like HIV, for instance, the mutation rate on HIV genome is very high. So it's evolving constantly. Okay. COVID-19 probably not as dynamic as HIV, but it does change occasionally over time. And so there are efforts, like there's a consortium of research institutions around the world, but most of it's being driven by one institution over in the UK, where they're taking samples from people that are positive, and they're actually sequencing the COVID-19 to look at those sequences and then to say, hey, this one's different by one nucleotide, one little base.
Yeah.
You can do sort of an evolutionary tree to look at it, and you can see where these are coming from and where the actual mutation even took place potentially. And so you can track that, and this is what people have done, is that they've tracked, okay, you had supposedly the original one in Wuhan, China.
Yep.
And it had a certain sequence, and it came over to the US. It also came over to Europe, but then also some changes happened in Europe, for instance, and then we noticed a different variant of COVID-19 moving over to the US on the East Coast, whereas the Wuhan one came over to the West Coast of the US.
Oh, really? Okay.
Yeah. But now it's sort of all mixing up because, we have just a lot of these social, basically, infections that are going on because people just are mingling, for a while before we had the lockdowns. And even now with the lockdowns, and we have people working on the front lines, in the hospitals and such, they're getting infected with many different ones out there now.
Okay. Let's see. In the future, is over-the-counter testing a possibility? And what do you think would be the implications of that? And is it something that's, if so, is it in the short-term future or long-term, or what's the situation maybe?
Well, it's a great question, interesting question. In fact, just today, I've gotten some emails. There's a group of us that have an email thread basically on the COVID-19 diagnostics, and one of the people on it used to be the head of the IVD, the Infectious Disease Diagnostics division of the FDA, and he basically he was there until about a year and a half ago or so, but he's still very much in tune with what's going on, and what he said is that right now there's no approved test for. There is one test where they've done some studies with, where basically the people can take the sample, but it's supervised. And it's supervised because taking a nasal swab sample is not trivial. It's not like just going into your nostril and just putting it in. So, and people probably it'd probably be hard for some people to even do what they're supposed to do properly to get the appropriate.
Yeah, I got that test, and it's not likely that I'm putting a Q-tip, that far up there.
Yeah, so you can imagine right now that that type of test would be probably pretty hard for a lot of people to administer to themselves at home. Okay, the serology test, the blood test for the antibodies, is something where you have to draw blood. Okay, and that is also something that's not trivial, and there's risks involved with that in terms of the possible infections and secondary things that could become quite serious.
Oh, okay, yeah, because I was picturing more of like when you do like like a diabetic would test their blood sugar, like that sort of a test, because that's when they did my antibody test, they pricked my finger, and they almost didn't feel it. So, but I don't know if if that affects the sensitivity or anything, or
well, yeah, it can affect the sensitivity because it's not clear right now, and more work has to be done. But it might be that at some point in time, if you might have in a drop of blood a sufficient amount of the antibody that you can detect, and also say that you have the right antibodies and the right amount of antibodies to be able to mount an effective immune response to a COVID-19 infection should you get it again, or should you be exposed again. So that may happen in terms of antibody tests. I think for nucleic acid tests, it's probably less likely in the near term. I know people are looking at other things like saliva. And,
okay.
And so that might be another approach that people will use, but these studies are not too far along yet, and so a fair amount of work needs to be done before that happens.
Now, it's been my understanding, this is just hearing, but it's pretty much, the reason why they go into the nasals is because that's kind of where it likes to hide out, is in the upper, or in the respiratory system. So, right. So would saliva be a good option, or how does that work? I guess would just be my.
Well, a lot of people, they basically breathe, not just through their nostrils, but through their mouth and things like that. So, and as you know, if you cough, it comes out through your mouth.
Okay.
Okay. So, and that, these micro droplets can come out, and they have the virus in it. Okay. And that's what causes these infections to go from person to person, or at least one mode of action to do that. And so the thought is that yes, saliva could have viral particles in there. It's just not clear how closely does that correlate to what's in your nostrils.
Yeah.
And what's in your nostrils, how well does that correlate to what's in your lungs, for instance?
Okay.
Yeah,
that totally makes sense. So in a case where somebody has obvious exposure, because this is kind of related to our situation, so a case where somebody has obvious exposure and doesn't get sick or even test positive for the antibodies, is there a difference? Is there any different immune characteristics that may cause this to happen, or is it test sensitivity, or just blind luck, or what's because I'm like, well, that doesn't make a whole lot of sense, and we're hearing this quite a lot, we're hearing like, just hearing that, oh yeah, these people were around all these other people that had this, and then they just don't have a sign of anything, not just symptomatically, but even through the testing.
Jason, that's a great question, and I think that's sort of the $64,000 question right now that we have out there in terms of what's going on there in terms of the why is it that you can cohabitate in a very small space with someone that is infected, and yet some people don't get infected. And I think the attention over the last few months has really been applied more towards identifying the people who are infected. Okay.
Yeah.
And now I think it's just like, okay, we need to understand, okay, there are people that have been exposed, and and probably, in a sense, constantly exposed for a significant period of time, yet they have no sign of disease. They have no sign of being even infected at all with antibody tests. So what's going on? So there are studies that are going on now that are looking at what they're calling susceptibility. Are you susceptible to this type of infection? And if you're not, what are the possible factors are there? Now, they could be genetic factors. They could be physiological factors. And, maybe if you're always exercising and maybe you have more ketones or something like that in your lungs or things like that. And,
Okay.
One interesting thing is like you make nitric oxide, for instance, NO, naturally. And there's evidence, although there are clinical trials going on now, that NO could potentially help with an infection in terms of helping your lungs reduce the effects of the infection. And it's even possible, and they're looking into this, that maybe NO might be something that could actually prevent infection. Okay.
Wow. So there's some supplements like I think it's L-arginine, and some other supplements that increase NO, and as well as things like nose breathing, which of course wouldn't be a good reactive way to go about things, because your nose is all stuffed up. You're not doing a whole lot of nose breathing, but is maybe a proactive thing that if done while doing cardio or meditation or something like that. That's just of course spitballing ideas with relationship to that.
It's sort of like certain people talking about the malaria treatment with quinacrine or something like that. The jury's still out to see whether there really is an effect, or maybe some people can benefit and others it may actually be more harmful to them than beneficial. So people are looking at all these different things to try to figure out what's going on and what confers some kind of natural resistance to infection, to COVID-19. And once, we have a better understanding for it, we might be able to test people, to see if they have natural immunity towards it, or natural resistance towards it. And, or, are there certain types of lifestyle changes, dietary changes, things like that might put a person in a better position to be less susceptible to infection. So,
okay. I was curious because we've been trying to talk about that quite a bit, because when I was a kid, I tested positive for TB. I never had it, but or at least I never had any symptoms or anything, but I tested positive for it. And so, we're wondering, TB is a bacteria, and this is a virus. So, but there was a couple things we looked at that, that was a very positive thing, like, oh, maybe it's that, maybe, your allergies actually created something, maybe, and then definitely lifestyle, being a kinesiologist, that helped improve, some of those other mechanisms. So, that's been a big point of curiosity, and wondering if, there was a, very underlying, an underlying biological explanation for any of that. But, sounds like they're great ideas, but not necessarily supported so far as of yet,
As of yet, it's in progress, that type of research, and trying to determine what those factors are. And, obviously, once they have a better handle on it, I think, they'll definitely publish on it, and then try to see what kind of interventions or prophylactic types of activities or foods or whatever it might be that could help in terms of resisting the infection. But, and obviously the other thing too is waiting for that vaccine to be developed,
Absolutely.
really effective. And I think once that happens, then people will be much more at ease in terms of being able to socialize in a much more normal way, even with the the local and federal governments later, at some point in time, easing the restrictions out there, I think there will be a fair amount of apprehension about going out there and doing things in public, and they probably will limit the amount of exposure and the distancing that you still have to maintain, and maybe even still wearing masks and all that, until we have something there that can better assure people that they're not very likely, or hopefully very unlikely, to get an infection, even if exposed.
Okay.
Okay.
Well, I mean, that's all I had. Is there anything else? I can't think of anything else. Those were the major questions that I had, and then it kind of come up. I mean, thank you very much. It was extremely informative.
Oh, you're very welcome, and I'm happy to answer any questions at a later date. Maybe in a matter of a few months, I might do an update or something like that and see.
Oh, that'd be great. That'd be terrific.
So.
Okay,
well, no, this is fun. It's fun. I think it's interesting. I think hopefully it's informative to those people that you interact with, and if they have any questions, I guess through you, they can ask me, and I'd be happy to try to answer them.
All right. Thank you so much, Carl.
All right, Jason. Take care, and I'll send you the link for the recording.
Oh, okay, so it'll just have like a link that I'll tap on, and then I can like download it from there.
You should be able to. That's my understanding.
Okay, terrific. Thank you so much.
All right, you take care.
You too. Bye.
All right, bye now.